The Cre-LoxP system is a technique used in a variety of research studies that allows for spatial temporal control over gene deletion, inversion, and translocation. The technique utilizes the Cre-recombinase enzyme to catalyze site-specific recombinations. Due to its efficiency in manipulating genes and chromosomes in a wide range of organisms, it is a powerful tool that is widely used in modern-day biochemical and molecular biological research. Previous genetic knockout experiments were restricted if the genes of interest were necessary for embryonic survival, or if the gene in question functioned in multiple tissues or cell types. Cre-Lox made it possible to conditionally knock out genes solely in subsets of cells where Cre-recombinase is expressed. Cre-recombinase is named because it creates recombination. It is an enzyme derived from the P1 bacteriophage and is designed to cut out genetic sequences between two LoxP sites. The Cre-recombinase enzyme contains an active site consisting of a catalytic triad with the residues arginine 173, histidine 289, and arginine 292, and nucleophilic residues tyrosine 324 and tryptophan 315. This allows for Cre-recombinase to bind to its substrate DNA. The name lacZ comes from the locus of crossover in P1 bacteriophage. lacZ is a site on the P1 bacteriophage consisting of 34 base pairs. This includes two symmetric 13 base pair sequences and an asymmetric 8 base pair sequence. Because of the asymmetry of the lacZ site, Cre-recombinase can either catalyze a deletion, inversion, or translocation of the genetic sequence between the sites, depending on the orientation of these lacZ sites. lacZ sites going in the same direction results in deletions. lacZ sites going in opposite directions can result in inversions. Interchromosomal recombination can also result in translocations. There are two main ways to get Cre-recombinase to be expressed in a cell. The first is to use a virus to introduce the gene for Cre-recombinase into the cell. The other method is to generate transgenic mice that insert the gene for Cre-recombinase after a specific promoter. Because this promoter can be expressed in certain cell types, the expression of Cre-recombinase can also be limited to these cells. There are many applications of Cre, one of which is lineage analysis, which is the determination of the origin or fate of cells. One example of lineage analysis was used by the Melton lab in determining the origin of adult pancreatic beta cells. They used an inducible version of Cre to determine whether new beta cells were derived from stem cells or pre-existing beta cells. In their experiment, transgenic mice were generated in which the insulin promoter drove the expression of tamoxifen-dependent Cre-recombinase, the enzyme that existed in the cytosol as a complex with the estrogen receptor and heat shock protein. When tamoxifen was injected in the cell, the Cre-ER complex separated, and the Cre-recombinase entered the nucleus to perform recombination. Once in the nucleus, the Cre-recombinase cut out the lacZ stop sequence, and thus allowed the expression of the HPEP label, short for human placenta alkaline phosphatase. Through this process, cells that normally produce insulin, namely the pancreatic beta cells, were tagged with the HPEP label. Using these mice, the Melton lab performed a post-chase experiment, the post being the set of cells labeled by HPEP using Cre-recombinase in the pancreas, and the chase being the set of cells that had the label after a given amount of time. Starting with a population of fully labeled beta cells, they hypothesized that if new beta cells arose from pre-existing ones, the percentage of stained cells wouldn't change. However, if they develop from unlabeled stem cells, the new population will have a much smaller percentage of labeled cells. Their data show that the percentage of labeled cells remained constant, which suggests that new beta cells were derived from pre-existing ones rather than from stem cells. Thus, the Cre-Lox system can be used for numerous types of modifications. Because it can be designed in many different ways, it has been used in a variety of different research studies that involve many different tissue types and organ systems. Perhaps in the future, this technology can be used for cures and treatments in humans. It has already been demonstrated that Cre-LoxP functions in human embryonic stem cells. There is definitely great potential for this system, not only in research but also in clinical settings.